Good day. Today we will be having our fourth journal review, and it is entitled Morpho-Physiological Responses of Pisum sativum L to Different Light-Emitting Diode, Light Spectra in Combination with Biochar Amendment. Presented by yours truly, Gilbert L. Florage. Here are the respective authors of this journal: Antonella Polzella, Mattia Terzaghi, Dalila Trupiano, Silvia Baronti, Gabriela Stefania Sipa, Donato Chiatante, and Antonio Montagnoli. And now, let us proceed to our introduction. Among the many requirements for plant growth and development, light plays a key role in different processes such as photosynthesis, plant defense, and phototropism. To provide the best fitness, plants developed morphophysiological strategies to use the available light conditions. Indeed, thanks to photoreceptors such as phytochromes, cryptochromes, and phototropins, plants can use light of different quality, quantity, and direction. Applications such as improvement of antioxidant activity in pea, growth in potato, and metabolism in mint, basil, lentil, primula, and marigold. Similarly, higher growth rates due to enhanced photosynthetic and transpiration activities have been observed in cucumber. Moreover, light affects root growth in grape plants and in seedling of different tree species. In addition, light modifies the transition of flowering, delaying or inhibiting the process in Indian mustard and basil, and promotes the suppression of anthocyanin content in lettuce. The effect of electrical light sources on plant growth depends on the properties of the light type used. The most efficient photosynthetic yield occurs in two distinct broad peaks in the blue, which has 400 to 500 nanometer, and in the red, it has 600 to 700 nanometer ranges. Photons in the 500 to 600 nanometer range have low photosynthetic efficiency, while photons in the far red, it has 700 to 800 nanometer range, are important for shoot elongation in woody plants, which is modulated by the ratio of red to far red light. In the present study, or here in our journal review, the response of the fast-growing model plant Pisum sativum L to different light spectra was investigated in combination with Biochar Amendment. We wanted to test the hypothesis that Biochar application could be used efficiently as a component of the soil-less container substrate, enhancing plant performance independently of the light spectra applied. To accomplish this, several morphological traits and physiological characteristics related to the photosynthesis machinery were measured in pea seedlings grown with or without Biochar supplement under three different LED spectra and one fluorescent light spectrum. And now, for continuing our journal review, let us proceed to the methodology. First, let's talk about the plant material and experimental setup used in this journal or in this study. Seeds of Pisum sativum L, which has a medium to late variety or medium height. Plant is long, dark green pod with 8 to 9 leaves and large wrinkled seed with dark green integument. It is proposed by Sementi Dotto, were obtained from a commercial nursery which in Varese, Italy. Pea seeds were sown one each in 2-liter sliced off inverted cone pots. It has a height of 15 centimeter, lower than 11 centimeter, and upper to 16 centimeter. It is filled with commercial soil-less substrate characterized by different proportions of composition in the case of biochar treated, which has 1 to 2 by 1 by 1 mixture of peat, silica sand, bark humus, and biochar, and control, which has 1 by 2 by 1 mixture of peat, silica sand, and bark humus. Pots were then placed in a growth chamber. 6 biochar treated plants and 6 non-treated plants were grown under each light spectrum, resulting in a total of 48 plants. From the 27th day after sowing, when the leaves of Pisum sativum were fully expanded, chlorophyll fluorescence emission and stomatal conductance were measured at 4 days intervals until the 43rd day after sowing. Seedlings were destructively sampled at 49 days after sowing for the analysis of morphological traits. All potted seedlings were watered to saturation with 400 milliliter tap water every 2 days. No fertilizer was added. For our second method, it is for growth room characteristics. Plants were kept in a single 4-meter wide, 3-meter deep, and 2.2-meter high growth room at the University of Insubria, Varese, Italy. The room was subdivided into 4 sections with reflective white panels. Each section was illuminated with fluorescent light. So here are the fluorescent light used, the Flora T8 Osram. And another one is the LED Vancy GmbH. It is from Garching, Germany. Or one of the 3 different commercially available is the LED light spectra. So this is the second light used in this study. It was developed specifically for horticultural purposes and coded as follow. So it is the code Valoja Oy, Helsinki, Finland. NSI is AP67 to 3L and AP67. Seedlings were grown under long day conditions, which is 16 height light over 8 height dark cycle, with lights turning at 10 a.m. Each section had a steel table with 50 mm high edges. Light intensity yielded approximately 250 to 300 mole. Light meter sensor used is HD 2302.0 Delta Ohm, which is from Castel di Selvazzano, Italy. At pot height, the plants were grown under 27 by 17 degrees Celsius day overnight temperature, with air humidity maintained at 60% to 70%. In our first table, you can see the spectral distribution and red to farad ratio of 3 LED light sources and fluorescent light. So in this study, they use a light source which is LED and fluorescent. And in continuous spectrum wavelength, there is 400 to 500, 500 to 600, 600 to 700, and 700 to 800. And the last one is the red to farad ratio of LED light. In LED light, they use 3. The first one is NSI AP67 3L. The second one is continuous LED. And the third one is AP67, while in fluorescent, they used Flora T8. And the numbers indicated in the table is the percentage of the continuous spectrum wavelength of the LED light and fluorescent light being used as a light source. And also the last part is the red to farad ratio of 3 LED light, which has in NSI AP67 there is 8.2, while in continuous LED there is 5.6, and in AP67 there is a 2.2 red to farad ratio, while in fluorescent there's a total of 5.7 red to farad ratio. For our third method, it is the biochar characterization. Biochar used in this study was produced by Romagna Carbone from Italy, from orchard pruning biomass through a slow pyrolysis process with an average residence time of 3 hours at a value of temperature in the middle of the production range, 500 degrees Celsius, in a 2.2 meter in a diameter and holding around 2 tons of feedstock. The pH was measured by potentiometry, which is pH meter, EUTech instrument pH 700 2013. According to the International Biochar Initiative IBI, Biochar Standards of 2014. The electrical conductivity value was obtained by the direct instrumental determination in 1 of 20 soil of water. It was according to the IBI standards of 2014. Cation exchange capacity was assessed according to Melich using BaCl2. Moisture content was calculated according to the Black method as the difference in sample mass before and after oven drying at 105 degrees Celsius to constant mass. In the present work, carbon stability was defined as the molar ratio of hydrogen to organic carbon, which has a maximum of 0.7 according to IBI standards of 2014. Total nitrogen, total carbon, organic carbon, and hydrogen contents were determined by dry combustion using a CHN elemental analyzer or Carlo-Erba instrument mode of 1500 series of 2. In the case of carbon, combustion was carried out after the complete removal of organic carbon with acid. Available nitrogen was determined by a modified Kjeldahl procedure using the Vardas alloy as reducing agent to convert NO3 and NO2 into NH4 positive and subsequent to Kjeldahl digestion. Total phosphorus content was determined by spectrometry UV1601 Shimadzu according to the last method described by Bowman. Available phosphorus was extracted by a NaHCO3 solution at pH of 8.5 and evaluated by spectrometry according to Oslen. The alkalinity of samples with a pH value greater than 7.0 was determined by tetraimetry according to Higginson and Ryment. The biochar tested was found to meet the European Biochar Certificate of EBC 2012 and IBI standard of 2014 requirements regarding carbon total and carbon organic content respectively. Its carbon hydrogen value was close to 0.7, ensuring good stability of the organic carbon. Conductivity values indicated that the biochar used has a high salt content. Moreover, available phosphorus and nitrogen represented 17.7% and 0.3% of the total phosphorus and nitrogen, respectively. Our fourth method is the substrate characterization. To assess chemical and physical properties of the substrate and the effects of biochar on these characteristics, 4 substrate samples were collected before and after biochar application and characterization for cation exchange capacity, total phosphorus, available phosphorus, total nitrogen and available nitrogen, and total carbon content as described in the previous paragraph. The pH was determined by potentiometry. It is by pH meter UTEC instrument pH 700 of 2013. According to Conyers and Davy, EC was measured by direct instrumental determination according to Ruades. It is the measurement of exchange capacity, while the different forms of available mineral nitrogen were determined by ion-selective electrodes on substrate samples dissolved in the ionized water. Next is the morphological measurements. Upon measuring of stem length, it is measured by centimeter by image acquisition of dissected stems and branches using a portable scanner, Epson Perfection V600. Images were then analyzed by ImageJ software. Leaf area was measured by scanning the leaves and subsequently analyzing the image by WinRISO. Leaf area was summed for each seedling to obtain total leaf area by centimeter squared. After gently removing each seedling from the medium, roots were rinsed repeatedly with running tap water and scanned by 400 dpi with a calibrated flatbed scanner coupled to a lighting system for image acquisition. Expression 10,000 XL, Epson America Incorporation, USA. Total fine root length was measured in centimeter, was obtained measuring only lateral roots by WinRISO excluding the taproot. Afterward, fresh biomass of leaves, fruit, and fine roots was obtained by weighing. Afterward, the plant tissues were oven drying for 52 height at 75 degrees Celsius and weighed again to obtain the dry mass of leaves, fruit, and the fine roots. Next is the physiological measurements. Chlorophyll fluorescence was measured with a portable pulse modulated fluorometer on the oldest fully expanded leaf randomly selected from each seedling. Measurements were repeated 5 times for each plant every 4 days starting from the 27th day after the sowing. The maximum quantum efficiency of photosystem 2 photochemistry for chlorophyll fluorescence in dark adapted conditions. F0 is indicating the minimum value of chlorophyll fluorescence obtained with the measuring light switch on. FM indicating the maximal possible value for fluorescence obtained with the application of a saturating pulse to a dark adapted leaf. V substrate indicating the variable fluorescence as the difference between F0 substrate and FM substrate was measured in the mid-morning when lights were still off or right after their turning on 10 to 11 a.m. In the latter case, leaves were pre-darkened with a leaf clip for 45 minutes to ensure complete relaxation of all reaction centers. For the statistical analysis, To evaluate significant differences between values, 3 comparisons for each morphological and physiological measured parameter were performed. First is the untreated versus biochar-treated plants grown under the same light condition. The second one is untreated plants grown under different light condition. And the last one is the biochar-treated plants grown under different light conditions. Morphological data were analyzed using a two-tailed t-test. Physiological data were analyzed by a two-way ANOVA with biochar treatment and lighting type as fixed factors and with time as a random factor due to repeated measures.